Okay, you can talk to student. Live now.
Okay, hello, good afternoon, everybody. This is the first time that I will be doing online class with Matthew course students, and I would like to welcome you all. Now, if you remember, the last topic that I discussed before the classes were canceled was the topic about Mendel's pea plant experiment. Now, several of the topics were already taken up in your Matayong 3 class. So I decided to proceed on to the next topic, which is nucleic acids.
Teacher, now we have students just 13.
Please, how do I do?
You have PowerPoint, right? That's fine.
Oh, this one. I cannot see so well.
Now we have 16 students come. You can look this one. And you can talk with students.
I cannot see so well, miss.
Don't worry, teacher, you just
Okay. Welcome to our first online class in science. And as I mentioned earlier, instead of proceeding with the lesson that we left before the school closed, I decided to proceed with the new lesson on nucleic acid. Now, for this topic today, I'm going to introduce you to the two types of nucleic acid, which is DNA and RNA. And then next week, we're going to do RNA only, and then the process of transcription and translation. Okay, let us move on. Now, before I proceed with the lesson, I would like to introduce some vocabulary first, okay? Because this vocabulary, you're going to encounter it as the lesson goes along. Okay, we have the first vocabulary, which is nucleic acids. Now, I am not sure if you've taken this up in your Thai science class, but in English, when we talk about DNA and RNA, we are talking about nucleic acid. This is the common term that we use. So, nucleic acids are large biomolecules. When we say biomolecules, these are molecules found in living things from the term bio. And this is the common name that we use for DNA and RNA. Okay, we have vocabulary number 2. We have the term synthesize. When we say synthesize, we are referring to combining things together to make something new. Like, for example, if you want to bake a cake, what do you put in the cake? Okay, we put flour, we put egg, we put sugar, we put flavoring, and so on. So, when you combine all of these things together to produce the cake, we use the term synthesize. Now, synthesize is also used in chemistry. Like, for example, if you want to produce other substances from different reactants, you say, we synthesize. Okay, let's say, for example, I have hydrogen and sulfides or sulfates. When hydrogen and sulfates are synthesized, we produce sulfuric acid. I'm sorry, I cannot do the shout out because I'm standing here and I cannot see from where I am. Okay, vocabulary number 3. we have incorporate. When we say incorporate, we are talking about taking something as part of a whole, or you include or integrate a part into a whole. So it means it's like something you're taking in something to become a part of something bigger. Okay, let's look at this one, incorporate. So we have here examples of the word incorporate as used in sentences. Number one, all words highlighted and underlined in red are all incorporate. So example, paints incorporate with oil. So that means that if you make paint, there has to be oil in it. Number two, many hotels will incorporate leisure facilities in the form of fitness centers or dance floors or both. Okay? So if you have the hotel itself, they will add leisure facilities in the hotel. And what are these leisure facilities? We have fitness centers and dance floors. Okay? So incorporate simply means to add to. Okay? Next, number three. Dress designs this season incorporate a variety of ethnic influences. Okay? So, if you're talking about fashion, the fashion this time will include ethnic influences. So, incorporate in simple terms means to include. Okay? To include in something or somewhere. Number five, we have the vocabulary strand. Now, students, all of this vocabulary that I'm introducing to you, you will encounter it throughout the lesson. So we have the term strand. To make it easy for you to understand, I use the example spaghetti. Or we can also use the example hair or thread. So when you say strand, it simply means things that are long and thin, like a rope. So here you can see several strands of spaghetti. A strand simply means one. Okay, one length of the spaghetti. That is a strand. You're going to encounter this vocabulary when we go to the parts of the DNA molecule. Okay, so we're done with the vocabulary. We're now going to proceed to the topic on nucleic acid. This is our lesson proper. Now, if you were able to bring home your books before the school's closed, you can find it in my World of Science, pages 55 to 56. If you have it, okay? If you don't have it, it's fine. Right. For students in rooms 1 to 4, I believe that you've encountered this term before, nucleic acid. As I've introduced to you in the early part of this lesson, we said that nucleic acids are large biomolecules. So, in short, again, these are biopolymers. Students, when you say polymers, this is something very long. Okay? DNA molecule is a biopolymer. So bio means life, and polymer means something that is very long. Or large biomolecules. As I explained earlier, biomolecules are molecules found in living things, both plants, animals, viruses, and bacteria. So essential to all forms of life. Now, there are two types of nucleic acid. The first one is the DNA, or the deoxyribonucleic acid, and RNA, or ribonucleic acid. I have here the function of nucleic acids. Now, for this period, for this lesson, I believe we cannot go to the second one, the RNA. I can teach you only DNA because it's only 50 minutes. Right, let's look at the functions of the nucleic acid. Number one, the storage and expression of genetic information. So, nucleic acids serves as a storage material of genetic information. What are these genetic information? These are the traits that you inherit from your mother and father. If you remember the past lessons, these traits include hair color, eye color, color of the skin, height, IQ, and so on. Okay. Number two, it encodes the information. This is for DNA. It encodes the information the cell needs to make proteins. So basically, the one that keeps all the information is the DNA, okay? And the one that does the protein synthesis is the RNA. But as I mentioned earlier, our focus is on this one only. So DNA stores the genetic material from the parents to the offspring, and then the RNA is the one that participates in protein synthesis. Okay, so I've introduced the vocabulary synthesis to you. Okay, let's move on. Now, how was the DNA discovered? In the beginning of time, man doesn't know anything about DNA, okay? But later on, because of research, it was first observed by a German biochemist named Frederick Meisser in 1869. That was the first one, okay? And later on, this model of the DNA was developed in 1953 by these people. We have James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. Now, this group of scientists figured out the structure of the DNA molecule, which is a double helix. I will show you later how the double helix structure looks like. So this is the first one, the one who initiated the discovery of the DNA molecule. This is Frederick Meisser. This is James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. So these people are the ones who did the research and the development of the DNA molecule. Now, what is the DNA molecule looks like? Again, students, I would like you to take note that the main function of the DNA molecule is to store genetic materials that you will inherit from your mother and father. Now, it's like people. We have parts. We have head, we have arms, we have legs, we have eyes, we have different body parts. The DNA molecules also have parts. So what are these? We have the nucleotide. And each nucleotide is made up of three things. We have the phosphate group, sugar, and the nitrogen base. Now, there are 4 nitrogen bases. The first base is called adenine. We designate with the letter A. Thymine, we designate with the letter T. Guanine, we designate with the letter G. And cytosine, we designate with a letter C. Now, I'm going to teach you later what are the base pairings for these nitrogen bases. So again, the DNA molecule is made up basically of the nucleotide. The nucleotide is made up of 3 parts: phosphate, sugar, and nitrogen base. And there are 4 bases. We have A, adenine, T, thymine, G, guanine, and C, cytosine. All right. So these are the illustrations showing us how the DNA molecule looks like. Now you will see here the codes, okay? Now basically, the DNA molecule is made up of 2 parts, 2 strands. Now if you cut the DNA molecule in the middle, you have this part, this is 1 strand, and this one is another strand. Now, what is a nucleotide? Now, if you look here, this is your nucleotide as we defined earlier. So, you have here the base, we have the sugar, and we have the phosphate. So, these 3 things make up 1 nucleotide. Now, we have the key here. So, for the key, orange is adenine, T, yellow is thymine, green is guanine, and purple is cytosine. Now, I want you to remember these base pairings. So, cytosine pairs with guanine, adenine pairs with thymine. Okay? Now, cytosine, guanine, adenine, thymine, guanine, cytosine, thymine, adenine. 1 strand to the right and another strand to the left. Now, these 2 strands are connected to each other by what we call hydrogen bonds. Okay? Now, why does it have to be hydrogen bonds? Because hydrogen bonds are weak. Now, during meiosis, when the cells divide, this DNA molecule will split. If the bond is strong, this molecule will not be able to split into 2. Take note that if it splits, the 1 strand left is what we call RNA. So it splits into half, left side RNA, okay, and this side is another RNA. So we have 2 RNAs, okay? So hydrogen bonds link the 2 strands together because hydrogen bonds are weak, okay? They are weak. Before I move on, students, I would like you to pay attention because when I give the final exam, the main topics covered will be this one and biodiversity. There will be 2 topics. So this topic, I will discuss it today and next week. And after next week, I'm going to move on to biodiversity. It's a lot of topics, so I will try my best to teach you as much as I can. Okay, so take note here, for cytosine, guanine and cytosine, you will see that there are triple bonds. Okay? And then thymine and adenine, there are double bonds. So during cell division, during meiosis, when these 2 splits, you have 1 strand, 1 RNA, and this side, another RNA. Okay, let's move on now. Now, this one here, sugar and phosphate, makes up what we call the sugar-phosphate backbones, okay? So this side is made up of sugar-phosphate backbones. And you will see here the base pairings. So sugar-phosphate backbone is made up of sugar and phosphate molecule. And then on this side here, this model here is what we call DNA ladder, look like a stair, okay? And then if we go here, this one is what we call the double helix structure proposed by the 5 scientists. So you can see it looks like this, okay? This is a double helix structure. Okay? Now, I'm sorry I cannot answer some questions because I'm standing up here and I cannot see well. Okay, let us move on.
Student just tell name and number and room.
Oh,
Just tell you.
because in M6 Miss, the students, yes, they ask a lot of questions in M6. Okay, students, let us move on now. Now, there are two classifications of bases. We have what we call purines and pyrimidines. Purines are classified because they have two circular structures. I will give you the details later. So the purines are guanine and adenine, and the pyrimidines are cytosine, thymine, and uracil. Okay? Now, uracil is not a base that is found in DNA. It is found only in RNA, and it replaces thymine. So, if you have the RNA molecule, you will not find thymine there because it is replaced by uracil. Okay? Can you remember? And you will see here that there is only one structure. Okay? One. That is pyrimidine. Whereas in purines, for guanine and adenine, you will see that there are two. Okay. Let's look now at the differences between purines and pyrimidines. For the structure, we have double carbon nitrogen ring. So, if you remember, two rings for purines and single carbon ring for pyrimidines. Second difference, we have the size. Purines are generally bigger in size compared to pyrimidines. Pyrimidines are smaller. And then source, where does purines come from? It comes from both DNA and RNA. So the purines are found in both DNA and RNA, but pyrimidines are found only in RNA. This is the one I was telling you earlier. Thymine is only found in DNA and uracil is found in RNA. Okay? Whereas cytosine is found both in DNA and RNA. So you have to remember that the only difference here is the presence of uracil in pyrimidines. It doesn't belong to DNA, it belongs to RNA. Okay, now this one is a brief review of our lesson from the very beginning about the chromosomes. Now, how is the double-stranded DNA molecules arranged? If you remember the structure of the cell, we have the nucleus where we can find the DNA molecules. Okay? Now, take note that the DNA molecule is a macromolecule, meaning it is big. So, it won't fit easily inside the nucleus of the cell. So, how do we find it in the nucleus of the cell? We find it arranged as chromosomes. So, chromosomes is made up of tightly packed DNA molecules. Okay? So, it is compressed, it is compacted, so it can fit inside the nucleus of the cell. Okay, so this is the one that I'm talking about. You have there the cell, you have the nucleus, and inside the nucleus you can find the chromosomes. And chromosomes are made up of tightly packed DNA molecules. So you will see here, this is the DNA molecule, and it is wrapped around the protein. what is that protein? We call it a histone, Histone. So the DNA molecule wraps itself around that protein and it exists in the nucleus of the cell as a chromosome. And the end part, the tip of the chromosome is called a telomere. Miss, I need to let them watch the video. Okay, you're going to watch a video about DNA, and we are going to answer some questions later.
DNA stands for deoxyribonucleic acid. It is a type of nucleic acid, which is one of the four major types of macromolecules which are essential for life, along with proteins, lipids, and polysaccharides. DNA. For deoxyribonucleic acid. It is a type of nucleic acid, which is one of the four major types of macromolecules which are essential for life, along with proteins, lipids, and polysaccharides. DNA is a double helix, which means that it is made up of two single DNA strands. Each DNA strand is in turn composed of many monomers called nucleotides, specifically deoxyribonucleotides. As a result, each strand is a polynucleotide. A nucleotide has three components: a nitrogenous base, a phosphate group, and a 5-carbon sugar, which for DNA nucleotides is deoxyribose, due to the lack of an oxygen group on one of its carbons. The sugar and phosphate groups alternate to form DNA's backbone. The phosphates act like connectors between the sugars by forming phosphodiester bonds between the third carbon atom of one sugar ring and the fifth carbon atom of the next sugar ring. Stands for deoxyribonucleic acid. It is a type of nucleic acid, which is one of the four major types of macromolecules which are essential for life, along with proteins, lipids, and polysaccharides. DNA is a double helix, which means that it is made up of two single DNA strands. Each DNA strand is in turn composed of many monomers called nucleotides, specifically deoxyribonucleotides. As a result, each strand is a polynucleotide. A nucleotide has three components: a nitrogenous base, a phosphate group, and a 5-carbon sugar, which for DNA nucleotides is deoxyribose, due to the lack of an oxygen group on one of its carbons. The sugar and phosphate groups alternate to form DNA's backbone. The phosphates act like connectors between the sugars by forming phosphodiester bonds between the third carbon atom of one sugar ring and the fifth carbon atom of the next sugar ring. These special carbon atoms are termed the 3' and 5' carbons. As a result, there is a directionality to this backbone. Each strand has a 5' end with a terminal phosphate group and a 3' end with a terminal hydroxyl group. There are 4 nitrogenous bases: adenine A, guanine G, thymine T, and cytosine C. Each of these attaches to deoxyribose via a glycosidic bond. The sequence of these 4 bases is the means by which DNA encodes genetic information, which can then be used to carry out all the cell's functions. The central dogma of biology describes how this genetic information is accessed and used by the cell. DNA is transcribed into RNA, which is then translated into proteins. The 4 nitrogenous bases can be divided into 2 groups. Cytosine and thymine are pyrimidines, while adenine and guanine are purines. Pyrimidines are 6-membered rings, while purines are composed of fused 5- and 6-membered heterocyclic compounds. You can use this mnemonic to remember that. Climbing the pyramid, George avoided 2 pursuers. The 2 single strands of DNA composing the DNA double helix are held together via hydrogen bonds, and they run anti-parallel, that is, in opposite directions. One strand is 3' to 5', while the other is 5' to 3'. Each nitrogenous base on one strand binds to its complementary base pair on the other strand. Purines hydrogen bond to pyrimidines. Specifically, adenine binds only to thymine via 2 hydrogen bonds, and cytosine binds only to guanine via 3 hydrogen bonds. Due to this complementary binding, each of the 2 DNA strands contains the exact same information. When the 2 strands are separated during DNA replication, each of them acts as a template which contains all the information needed for recreating the other. The 2 DNA strands can be separated or melted in several ways: through mechanical force by enzymes, through high temperatures, or through extreme pH levels. Going back to the central dogma of biology, DNA gets transcribed into RNA, which then gets translated into protein. However, not all sequences of DNA are actually meant to be transcribed into useful RNA sequences. Those sequences of DNA which are actually meant as a template for transcription of RNA, which then gets translated into functional proteins, are called non-coding or antisense sequences. The DNA which is complementary to an antisense sequence is termed a coding or sense sequence. At first, it may seem counterintuitive that the strand which is used to transcribe RNA is called non-coding, but note that this transcribed RNA is complementary to the non-coding sequence, and so it exactly matches the coding sequence. Note that both strands of DNA can have sense and antisense sequences. Furthermore, sense and antisense sequences can overlap. For example, there can be overlapping genes where one protein is encoded on one strand and another is encoded on the opposite strand by the sequence read in the opposite direction. Since transcription can only proceed in the 3' to 5' direction along the template DNA strand, the RNA being synthesized is always 5' to 3'. Thus, a sense sequence, for example CAAT, will never be transcribed, although if the reverse sequence TAAC is an antisense sequence, it can be transcribed. The twisting of the double helix creates several more interesting features in DNA. Firstly, the grooves formed by the twisting are not equally spaced. One of these grooves is called the major groove, which is 22 angstroms wide, while the other, the minor groove, is only 12 angstroms wide. These grooves often provide good binding sites. In its relaxed state, each DNA strand makes one loop around the axis of the double helix once every 10.4 base pairs, with a pitch of 34 angstroms. If the DNA is being twisted tighter in the same direction, this is called positive supercoiling, while being twisted in the other direction is called negative supercoiling. In eukaryotes, DNA is found in the nucleus, but in prokaryotes it is found in the nucleoid. In both eukaryotes and prokaryotes, the DNA is packaged up into chromosomes, which are a combination of DNA and proteins. Chromosomes make the DNA more condensed and organized, and allow the cell to have controlled access to specific portions of DNA. Eukaryotes have linear chromosomes, while prokaryotes have circular ones. The genome is the complete set of chromosomes in a cell. If you enjoyed this video, like and subscribe. You can also support me by following the link to my Patreon.
That one. Okay. All right, students. I have here some guide questions for you to answer, but I really cannot see your answers now. So I think I have about 10 of it. So the first one is, what is the meaning of double helix? Okay, you will see that several of the vocabularies that I introduced to you earlier are all here. What is the meaning of double helix? Double helix means two strands of DNA that are intertwined. Okay, that is a double helix. Next, number 2. What are DNA strands made of? DNA strands are made of nucleotide. Okay, they are made of nucleotide. Miss, do we have a pen? This one?
Yes. You can write on the board.
On the board. Do I need to use anything? Any pen? Okay, thank you. Okay, let's try number 2. Number 1. What is the meaning of double helix? Double helix simply means two DNA strands. Okay, two DNA strands that are intertwined. Next, number 2. What are the DNA strands made of? It is made up of what we call nucleotides. Okay, nucleotides. And the 3 components of a nucleotide are what we call base. So I have the base, sugar molecule, okay, bases, and we have the phosphate molecules. Okay, so these are the 3 components of a nucleotide. Next, number 4. The DNA backbone is made up of what types of molecules? Now, basically, there are 2. The backbone is the side of the DNA molecule. If you remember the ladder structure that I showed you earlier, there are rows from top to bottom and then the sides. That is the DNA backbone. Basically, that one is made up of sugar molecules and phosphate molecules. Okay? So this is the basic parts of the DNA molecule. Let's move on to number 5. Name the 4 nitrogenous bases. Now, I'm not going to write the full name here, but I'm going to write the letters that symbolizes them. So we have adenine, guanine, cytosine, and thymine. Okay, adenine, guanine, cytosine, and thymine. Now, what is the difference between purines and pyrimidines? So, we can say that purines is made up of 2 carbon, nitrogen structures, okay? Whereas pyrimidines have only 1, right? Now, number 7, what holds the 2 strands of hydrogen bonds together? Sorry, I think this one is not correct. Oh, okay, number 7 is not correct. So what holds the 2 strands of not hydrogen bonds, the two strands of DNA. The two strands, sorry, what holds the two strands of RNA together? The answer is weak hydrogen bonds. Okay, weak hydrogen bonds. Why do we have to call it weak again? Because during the process of cell division, when the DNA molecules need to split into two strands, the bonds that hold the two strands together need to be weak, so it's easily separated during the process. Okay, let's move on. Number 8, how do the two strands of DNA molecules move? How do they move? According to the video, they move in opposite directions. Okay, they move in opposite directions. Next one, number 9. What are the 3 ways by which DNA strands can be separated? All these questions, you can find the answer from the video. The first one is through the action of enzymes. Action of enzymes. And then number 2, we have extreme temperature. When I say extreme, I'm referring to high temperature. Extreme temperature. And number 4, high pH. Okay. You know, pH is a measure of the acidity or basicity of a substance. So you try to remember, DNA is made up of 2 strands. These strands need to separate during the process of cell division. And these 3 methods can separate the strands together, okay, by breaking the weak hydrogen bond. Okay, number 10. What is the difference between transcription and translation? In transcription, the DNA molecule splits into 2 strands. Whereas in translation, the one that works is your RNA. So RNA is involved in protein synthesis. Okay, that is the difference. Now, this process of transcription and translation will be discussed next week when we do the topic on transcription and translation. Okay, now these 2 will be discussed later next week. So, thank you so much for joining me during this live on-screen class in science. And then I will see you again next week, everyone. Bye. Thank you, Miss. You stay here every day? Every day.